Hi guys, sorry I can't be there today. On Friday we're going to be doing a really cool lab, so I wanted to do one of these weird screencasty things so that I can explain to you what we're doing, so you don't miss up on any lost time just because I'm sick. Um, Let me think. You should have already read the first 3 pages in your lab manual, and hopefully you're watching this video second. It's okay if you're watching it first on Educated. I'm just going to explain a little bit about sickle cell in this video, but more about the actual experiment that we're going to be doing on Friday. As you read, sickle cell disease is caused by the sickle shape of red blood cells. In case you don't know what a sickle is, that's what a sickle is. So it takes this weird elongated shape. On the right here we have a normal red blood cell. It's inflated, kind of donut-like, and nice looking. Obviously you can imagine some kind of problems that could arise with this shape. You could think about how it cannot carry oxygen, so that results in shortness of breath. Also, because we need oxygen for cell respiration, it's hard to produce ATP, leaving patients fatigued with sore, tired muscles, and often jaundice, which is a symptom with the yellowing of the skin and eyes, just because oxygen is not getting where it needs to go. There's lots of other symptoms of sickle cell, but I just want you to think about how this shape could affect the blood and the body. There's a sickle again, sorry. Okay. What causes this shape? That is this protein, hemoglobin. I'm sure you've heard of it before. It's the protein in red blood cells that carries oxygen from the lungs to the tissues and carbon dioxide from the tissues back to the lungs. Basically, hemoglobin is doing all the work for the red blood cells. It's actually carrying the chemicals, those molecules of oxygen and carbon dioxide, where they need to go and when. In sickle cell anemia, hemoglobin does not do what it's supposed to do. In a normal person on the left, hemoglobin takes this nice shape. It does not clump together. They're all individual proteins doing what they're supposed to be doing on their own. However, in sickle cell hemoglobin, the protein molecules clump together and make like this weird chain shape. They get stuck on each other. The reason for that is the actual shape of the hemoglobin is different compared from the normal to the sickle. This change is caused by one base pair, an A that somehow became a T. It's really hard to write with your mouse. An A that became a T in a mutation. So one letter was mutated and the entire shape of hemoglobin switched, like so. Because this hemoglobin has unfolded a little bit, the sickle hemoglobin, that's what causes these long chains of hemoglobin and ultimately this sickle shape of the cell. Okay, here we are. So why are we even talking about all this? Hopefully you already watched this video, so I'm going to skip it. Oh no, there's a lot of ink on this slide. Hopefully you also already solved this pedigree as well. I'll just explain these answers. The patient is right here. The patient, his uncle over here, and his grandmother all have lowercase s as their genotype because they have sickle cell anemia. It is a recessive trait, as you read in the article. With that information, you can kind of figure out a few other individuals. Unfortunately, not everyone can be solved here. Because the patient has two small s's, one had to come from mom and one had to come from dad. However, mom and dad look normal. So that means they have a dominant allele to kind of block that out. I use the $sign as dominant so that you can tell the difference between my s's because I'm messy and I can't write well. So using that information, the patient's genotype, you can figure out his parents, even though they're both normal. You can use the same method to figure out the grandparents, which are here and here in generation 2. using the patient's uncle as kind of your clue, we know that one of these little s's would come from the grandmother because they both have little s, little s. However, the other one had to come from father because remember, again, everyone has two alleles, one from mom, one from dad. So this small s here had to come from dad if this one came from mom. Again, dad presents as normal, so that means he has a dominant trait to mask that one. Same applies for the great-grandparents. You cannot determine these, I guess they would be a great aunt and uncle of the patient, and you cannot determine the patient's siblings. The reason for that is you don't have any information about their kids if you were to do like this, you know. So you don't know, you can't tell if this person is homozygous dominant or if they are heterozygous. Ooh, that's messy. There's no way for you to find out with the information given. So you can just leave a question mark or leave it blank. That's fine. Okay? So what are we doing in our lab? Every group, it'll probably be groups of 3 in our class, are going to be given a sample of a patient's hemoglobin, purified hemoglobin. It is not a blood sample. They are not blood cells. It is just the protein hemoglobin. Your job will be to figure out whether or not the individual has the sickle hemoglobin or the normal hemoglobin. How are we going to do that? Well, the overall charge of the normal hemoglobin will give us a clue. Because we won't be able to really zoom in and see the shape because of how small these proteins are, but we do know that normal hemoglobin has an overall charge of -2, this whole molecule. Just based on electron sharing, general bonding between, within the molecule. That is the overall charge. That's a given. In sickle hemoglobin, this one change, this green, indicates where that amino acid change occurs. Although it was one amino acid that changed, it changed the whole shape of the molecule, and it also changed the charge. The charge of sickle hemoglobin is -1. That is going to be the key to solving what your mystery patient has, normal hemoglobin or sickle hemoglobin. Here's how we're going to do it. Gel electrophoresis. What the heck is that? Well, let's find out. Start with the word gel. Here are all the things we're going to use. I'll get back to that. The gel is the first step to this lab. I will be making the gel for you, but I still want to explain what it is, where it came from, so that you can kind of figure out what's going on. The gel is made from something called agarose, which is not easily readable there. Okay. Agarose ends in ose. Ose. What's that? Hopefully you should say sugar. Or a carbohydrate, excuse me. Agarose is a sugar. So it starts out as like a powder or like grains, like regular sugar at your house. I added boiling water to it and mixed it up. And then I poured it into this like casting tray, kind of like a mold. And I put these combs in it. It's hard to see, but these combs have teeth, just like a regular comb that you brush your hair with. And I let it harden. This gel is a lot like Jell-O. If you've ever made Jell-O at home, this is how you do it. You buy the Jell-O mix. It's basically sugar, add boiling water to it, let it cool, and it will become hardened. The comb creates these wells in the gel, like little divots. Those are going to become very important. Okay. I'm going to leave an example of the gel up at the front of the room next to the sink. Feel free to go ahead and touch it and see what that looks like. Just don't stab it or break it. Thanks. Okay, what are we doing next? What are those wells for? The wells are where we're going to actually put our samples. So we're going to use this cool thing called a micropipette. I'm going to demonstrate that in class for you on Friday, but it's a really fancy pipette. We're going to load 3 samples into the wells per group. One's going to be the hemoglobin, which is going to do HB. Oh my god, that's a B, sorry. HbA. Oh, that's so ugly. HbA is the normal hemoglobin, the -2. You will also have an HB, hemoglobin. S stands for sickle, so you'll have sickle hemoglobin. And then you'll have your patient, which is a big mystery. Ooh, what does our patient have? I'll give you a hint, the patient is not always the patient from your packet, because he has sickle cell anemia. You don't know what you're getting. There's all different patients. Excuse me. You're going to load each sample into its own separate well. Okay? Next step. That gel with the samples in it will be completely submerged in this gel box, which will be filled with a buffer. The buffer is just a solution that conducts electricity a little bit better than water does, which is going to help us because we need to get electricity through the gel box. Okay? You are going to put your gel in the gel box, cover it with buffer, so that your wells are here. So all your protein are lined up on this side. I don't know if you've ever had to help a parent jump a car or messed around with wires for any reason at all. But you may already know that in electrodes, black is always negative and red is always positive. Okay? It's important to know. Life lesson if you ever have to jump a car. We are going to align our wells with the negative end. Okay? This is very important. Then we're going to we have a power source. They're around the room. We're going to connect to our gel box. So we're going to hook up our our negative and our positive end to a power source. I want you to think carefully about this. Here we have our proteins all lined up in these wells. Okay? Next to the negative electrode. The proteins are negatively charged. When we turn on the power, start supplying a bunch of electricity through here, what's going to happen? Think about how negatively charged, if you put two negative ends of a battery next to each other, what happens to them? Okay. You probably said they're going to repel. You would be correct. The proteins are going to repel against that negative charge from the voltage and are going to migrate through the gel. The reason why they can do this is the gel is like SpongeBob. It has a bunch of holes in it. They're definitely not that big. They're so small, they're like microscopic. But it's made up of a bunch of holes, like air pockets, and the proteins can actually travel through the holes, which is pretty cool. We have soaked them in blue dye, so you'll actually be able to watch them. It'll look like this. It'll look like a blue band. I know that's red, but whatever. And it'll travel along through the gel. Keep in mind that one of these is a charge of -2, whereas the other one is a charge of -1. I'm about to sneeze. Hold on. Ooh. All right. One of your samples is going to have a charge of -2, the other of a charge of -1. So when we look at our gels, the -2 is going to have a stronger affinity. Ooh. It's going to have a stronger affinity to go towards that positive electrode. Opposites attract. It's -2, it's twice as strong as the -1. Okay? So ultimately, when we're done, when we turn the electricity off after probably 20 minutes, we're going to see that the normal one will have traveled further through the gel than the sickle cell. Those who are heterozygous, so that's what that means, have the sickle cell trait, they can produce both normal hemoglobin and sickle hemoglobin. So you will see two bands. Sometimes it's kind of like smeared. It looks like one long one. This is your expected results. So these are those wells that we started with.